Good evening to all of you. So today we would like to extend some of discussion and thoughts on cancer, metabolic adaptations, and sometimes we refer as tumor metabolic heterogeneity. That means one type of the cancer will have different metabolic adaptations and their own metabolic pathways compared to the other cancer types. And also within one cancer types, one cancer cell compared to the other cancer cell among the billions of cancer cells will have its own metabolic signature or identities. So in this direction, there are many publications are available. Various understandings are available. However, today I would like to share insights on how the cancer cell and this cancer cell in a very simple way is mentioned as microcellular factory. Means micron in size, that's why microcellular factory in the form of cancer cells within a tumor. In other way, in a tumor, there are billions of cancer cells. All the cancer cells, they are behaving like a microcellular factory. And this microcellular factory in the form of cancer cell, they try to play around according to the growth conditions, whether under stress or under cell death and many more environmental conditions. So even cancer cells, they go for adaptations. And during that, what the whole idea is, this microcellular factory may be able to manage efficiently in a such a way this nano metabolic pathways that can be in the form of glycolysis, it can be in the form of TCA cycle, it can be in the form of polyamine metabolic pathway. This can be in the form of sphingolipid metabolic pathways, right? So these nano metabolic pathways are going to be efficiently managed. The whole understanding on the efficiently managed means some of the selected metabolic pathways are going to be shut down and some of the metabolic pathways may be elevated. So the cancer cell go for this metabolic adaptation by very efficient mechanism by controlling this nano metabolic pathways, right? Specifically today, I would like to discuss under condition of like stress or cell death under the effect of like chemotherapy drugs or radiation therapy or many more, right? So we will start with one proposed model. So what? So what is the whole point to discuss here? That cancer cell, it's a definitely microcellular factory, are very good micromanager. Correct? So the micron size of the cancer cell are micromanager during the stress and the cell death. And how these cells are working as a good, efficient micromanager in a normal growth settings where the very good condition of growth like nutrient, divalent cations, and many more things are available in the surroundings of the cell. So the cell will go for proliferation, invasiveness, metastasis, and many more things, right? So this efficient management of these metabolic pathways or cellular process during all this growth condition is known, right? We know cancer cells are managing things. However, and these cancer cells are highly plastic in nature, correct? Fine. Now, if you look at other way around here, this one, once the cancer cells are under stress or under cell death condition where the anti-cancer drugs are insulting upon the cancer cell, So the cancer cells are going to work even better micromanaging. So the cancer cells are going to possibly shut down certain metabolic pathways. Maybe they are not interested anymore to create certain new metabolites. And those metabolites are accumulated inside the cancer cell or at the intracellular levels. Those metabolites needs to be secreted outside, right? In the ECF. That can be one of the micro management by the microcellular factory of cancer cell during stress and cell death, right? Now we need to extend this particular proposition in this form. So whether the cancer cell during this micromanagement of these metabolic pathways, they are going to secrete or export, look at here, this one, export or secrete certain highly accumulating metabolites to the outside, that is in the ECF, right? No? So these cancer cells during the stress and cell death, so what we think, I mean, there are multiple propositions and possibilities are there. So during this cell death and stress, these accumulating metabolites inside the cell may be toxic or may be inhibitory in nature, right? So these metabolites, those are accumulating at the intracellular level that has to be secreted outside, one. Then second possibilities are there, these chemicals, those are going to be secreted outside. These metabolites may serve as a messengers or signaling messenger to the surviving cell. That is important point. So the cells, those are under stress or cells, those are going to die through the like cell death mechanism, like apoptotic cell death you can consider. So this dying cancer cell will ensure some of the metabolites are secreted outside as a messenger for the surviving cells. And then third proposition or possibilities are there that these secreted metabolites, not only the signaling messengers, they may also work as a oncometabolites, as a resource for further growth and proliferation or survival of cancer cells. So everything is happening in a tumor environment. Once this tumor is exposed to the chemotherapy drugs or radiation therapy or many more external insults, right? So based on this understanding, what we believe that the cancer cells are good micromanager to manage their own metabolic pathways, either by shutting down certain specific metabolic pathways or to elevate certain metabolic pathway. And most importantly, how the cancer cells are going to make a decision which type of the metabolic pathways should be shut down first, then in second, and then in a third priority. That is also very complex biological phenomenon in cancer cell. That is why cancer cells are referred as super complex micron size of the cell system, right? Now, the last point to make a relevance here, we have also observed in our own experiment and in the data that those cancer cells are under stress in a certain specific cancer cells. Some of the metabolic pathways, and I would like to highlight here one of the pathway that is sphingolipid or the sphingolipid metabolic pathway. So the sphingolipid metabolic pathway, we believe that during the cancer cell or under stress, these pathways are going to be micromanaged by cancer cells, right? So this pathway, if they are going to micromanage with cancer cell, how? So what we believe that in the sphingolipid metabolic pathways, the thumb rule is here. The sphingosine, if you look at here, is going to be phosphorylated inside the cancer cell to create a certain messenger metabolites, and those are going to work for growth, proliferation, and metastasis of cancer cell. But if the cancer cell are under stress or under the influence of anti-cancer drug, so the cancer cell may prefer to go for shutting down these sphingolipid metabolic pathways. And in that direction, the possibilities are there. Those metabolites of sphingolipid, and specifically I would like to mention here sphingosine, that is the precursor of majority of other sphingolipid metabolites. The this sphinge-9 is not going to be further metabolized. That means the whole metabolic pathway to involve this sphingolipid, the sphinge-9, is going to be stopped or shut down. And what will happen then? So then the accumulation of the sphinge-9 will be inside the cancer cell, and the cancer cells are under already stress or under the influence of the drug. So the best strategy will be to secrete sphinge-9 to the outside, that is the ECF of the dying cancer cell, right? And these sphinge-9 will be secreted to the outside, and then the other functionalities of sphinge-9 will be achieved by the tumor environment. Now, the important point is, if the sphinge-9 is not secreted outside, that means if the sphinge-9 is going to be accumulated inside the cancer cell, and those cells are under anti-cancer drug effect, then what will happen? The possibilities are there. This sphinge-9, if I remove the annotation, this one, this metabolites, if it is accumulated inside the cell, possibilities are there. And there are certain evidences we have also collected in our lab that sphinge-9 may be able to inhibit certain oncoprotein or oncoenzymes like protein kinase C, or you can say SRC kinase, etc. And that is not going to be in favor of cancer cell, right? So ultimately, the cancer cell are going to look at or the micromanaging at both front. One is if the metabolites are not useful, the whole metabolic pathway has to be shut down, and also that to avoid the accumulation of metabolites like sphinge-9 inside the cancer cell is not favorable for cancer cell, right? So those like this sphinge-9 has to be secreted outside of the cancer cell, right? To avoid further complications for cancer cell itself. So what we believe that this micromanaging by cancer cell at the nano metabolic pathways are very critical for cancer cell. That is why cancer cells are highly regarded as super complex and super efficient type of cell in comparison to the normal cell or healthy cell. So I believe that this information will be useful to all of you. Thank you very much for listening.